Topic is delineated molecular interaction mechanisms in vitro microbial plant communities. As plant microbe communities has been a well-discussed topic so far in the last couple of days, I want to focus mostly on our analysis pipeline, our modeling approach to identify these molecular mechanisms. We know that forests, terrestrial forests, are an important part of the terrestrial carbon cycle. Aspen forests are particularly interesting because aspens are very common in North America. They are highly genetically diverse. That genetic diversity maps to differential responses to elevated carbon dioxide in the atmosphere, and it's a sequenced genome by JGI. We also know that these communities are not standalone. The phenotype of these communities are directly dependent on their subsurface communities. So the challenge is to understand how these communities are going to respond to elevated carbon dioxide. We need to understand the community interactions between the trees, the roots, the soil fungus, and the soil microbes. Our goal is to model the specific molecular mechanisms that mediate the response, soil microbe community, to their environment. An in vitro model of this system gives us a tremendous power to dissect these interactions in a very tightly controlled manner. The organisms in the model I want to discuss today includes the aspen tree, the ectomycorrhizal fungus Leucaria, 4 flavors of mycorrhizal helper bacteria, 4 strains of Pseudomonas florensis. This simple model lets us mix and match community members, have an absolute control over the media matrix, and measure very specific phenotypes of the resulting aspen trees. At our current state in the lab, to dissect these interactions as carefully as possible, one of the things we have done is we're just setting these things up in their pairwise components to isolate the mechanism by which these things interact. So we have a fungus-bacteria communities, we have the plant-fungus interactions, plant-bacteria interactions, and we have found combinations of these communities by which the phenotype of the community is dependent upon the culture condition and on the media. So these things, the response of these things to another is dependent on the media environment. For the rest of the talk, I want to just focus on the plant-soil microbial combinations. The specific experimental design I'm presenting today grows the plants and the Leucaria together. In this case, the fungus is sitting on a membrane, preventing any physical contact between these two organisms. Isolating, so any interaction between these two has to be by those small molecular signals that can diffuse through that membrane. We have a time course of this experiment. We have taken short-read RNA-seq data for every point along this way. And we have a theoretical model for how we think these things interact. This is the chassis on which we're going to hang all of our subsequent analysis. The specific molecular mechanisms by which specific signaling molecules in the extracellular environment are detected by transmembrane receptors. The information of that protein ligand binding is relayed to the nucleus by a signal cascade through specific physical changes in these cascade proteins. The result of that is to change the binding activity of a set of transcription factors. The transcription factors bind to characterized DNA motifs. These motifs drive co-regulated gene clusters. The observed phenotype is a function of our change in expression. Because this spans multiple kinds of interactions, we need a multi-omics approach to this. So not only do we have the transcriptomics component, we also have a metabolomics component, a proteomics component, and thanks to JGI genomic sequencing, we have a genomics component, so we can search upstream of differentially expressed genes for specific transcription factor DNA binders. So the first component I want to discuss very briefly. So in the transcriptomics, we're looking at collecting the gene expression data from the metatranscriptome in these experimental sets. In the Aspen root, just group gene expression into patterns of co-expression, and we can associate these specific gene patterns with enriched function of gene annotations. For the metabolomics, the metabolomic component of this model is the signal molecule synthesized by the Leucaria. So from the Leucaria transcriptome, we build a metabolomic model using expressed enzymes and known metabolic pathways that we've extracted from KEGG. This is something that I get a lot of questions from. Some questions came from the poster. So just to give a slight metaphor for what this model calculates, this is not a flux balance model. This does not consider enzyme rates. In this model, which is a cartoon, the nodes are metabolites whose presence are inferred by the expression of specific enzyme activities. Edges in this figure are enzyme activities. I select one transcriptome to use as a reference. And if you can imagine this model, say metabolites are ping pong balls and edges are springs, for our control system, we set every spring length to 1. We look at differential expression in different communities, and we compare them to our reference. So if the gene is upregulated, we tighten that spring a little bit. If the gene is downregulated, we loosen that spring. If we let these springs reach a stable configuration, the ping pong balls that are our metabolites will have slightly different positions based on the tension of those springs. The distance that those ping pong balls move from one metabolome to another is the predicted relative metabolic turnover. And this turns out to be a very quick and dirty way to do some fairly interesting metabolic modeling, knowing very little about the enzyme kinetics of these systems. For proteomics, so far as I know, there is not currently available a large database of Aspen biologically, experimentally confirmed protein-protein interactions. So to identify potential protein complexes in this set, we first extract those genes that are statistically significantly expressed in our biological experiment. We take a subset of those that are annotated by JGI as likely components of a sensor complex. We pull out from all available Aspen root data, all of the available transcriptomics data, and we find which of those expressed genes are always co-expressed when we see them in roots. The assumption is if these elements form some components of sensor machinery, when we see one component, we should see every component. Since there is not a wealth of Aspen protein-protein interaction data, we use a database of Arabidopsis protein interactions. We find likely pairwise interactions statistically enriched in the Arabidopsis by their predicted subcellular localization predictions, and we identify a expected whole network topology for what kind of shape these kind of interactions are expected to take. And we find the overlap between these predicted complexes and those genes that are differentially expressed. So here we have the highlighted nodes are proteins in a complex that are also differentially expressed. And trivially, I'm just going to point out that these genes are very non-randomly distributed. There are some complexes that not only are comprised of things that are differentially expressed, but differentially expressed in statistically significant relevant patterns. For the genomics component, we look for enriched transcription factor DNA binding sites up to 1,000 base pairs upstream of the differentially regulated genes. 2 ways to identify these. The first is to look for known transcription factor binding sites that are statistically enriched upstream of genes within a co-regulated cluster relative to the distribution of DNA binding factors in the complete set of differentially expressed. And we look for things in those predicted complexes, which of those transcription factors in the predicted complexes have expected DNA binding sites. We can use this to assemble a complete model of our interaction system. So we have
specific signals, specific receptors, driving particular activities of transcription factors via these signal cascades. These are the predicted binding factors driving clusters that have particular predicted phenotypic responses. Is Sarah still here? Sarah, she presented earlier. She is going to get us to lunch a little bit quicker by having given this figure already a little bit, in that we have in the predicted figure components like salicylate, jasmonic acid, driving things like hypersensitive response for gene clusters that are defense response and are things like cell death. So this very naive, data-driven approach overlaps what we expect to see in these kinds of sensor apparatus. This ceases to be interesting. But one of the things we can also do is find the overlap between our predicted networks, and here's an example that was culled from KEGG for symbiote recognition pathways. In this figure, too small for you to see, but I just want to point out things highlighted in green are those metabolites that we identify by metabolic modeling. Red ones are those in our predicted sensor complexes. Blue ones are in our differentially expressed genes. So our very naive, data-driven model of symbiotic detection, again, overlaps a great deal of what we expect to see. The advantage of this kind of approach is those models produce sets of pairwise protein-protein, protein-ligand, protein-DNA interactions. These are the kinds of predictions that we can use to drive very specific biological experiments. So the result of this is not just a model, is not just overlap with what we already know. This defines a biological space that we can explore to confirm these models. Up to this point, we're getting to lunch early. Up to this point, we've only looked at single component models. One of the attractive ways of that fairly simple sensor complex is we can use them, arrange them end to end, such that the phenotype at the terminus of one model can be used as the signal components of the next model. So we can line these things up to generate a far more dynamic model. We can close these loops, bring them around, make this a truly dynamic system to identify how these things are communicating and how that communication further feeds into community interaction. Where do we fit on, I'm sure we've seen this a couple times so far, on the DOE scale of carbon cycle? The most elementary use of this kind of model is to identify molecular targets for characterization. The outputs of these models are predicted pairwise interactions. These can be validated, and this is driven by the predictions of the genome annotation. A little more exciting is understanding of these molecular targets open up the opportunity for eco-engineering of these communities. We want to drive terrestrial communities towards a favored carbon storage. It is probably a matter of centuries if we're thinking about reforesting with aspen trees that have a favored carbon dioxide response. It might be much quicker if we try to manipulate the microbial communities of these systems that might be a draw for a carbon sink. The last way, going all the way to the top here, the incorporation of these plant soil community systems can be incorporated into global system models of carbon turnover. And one of my hobbies is extrapolating to absurdity. So this particular example combines the community Earth system model data with the Earth microbiome data by way of the models that I've just presented. And so this is a prediction for every terrestrial environment microbial community of the relative carbon dioxide metabolism based on the Earth system model data. And then we've extrapolated this out to 2,100, which is an interesting computational exercise, more relevant to this system. One of the components of the Earth system model is the carbon input from the plants. Incorporating these interactions into things like Earth system model is going to give us better pictures of carbon model, how carbon atmospheric changes is going to affect soil metabolism over the next 100 years. I'd like to acknowledge collaborators here at Argonne, particularly Frank Collard, Sherlocka, at the University of Alabama at Huntsville. This is the lab which generates a number of the physical plants in this system, Leland's lab in Huntsville. Thanks to Sarah, we finished a little early, and I would be happy to take any questions.
The model results are impressive to go from those stages, from one stage to the next there. I'm just wondering, there seems to be more impressive to me because some of the assumptions that have to go behind it when you're modeling an ectomycorrhizal system on protein-protein interactions with Arabidopsis, and which isn't an ectomycorrhizal host, right? And then some of the fluorescent, Pseudomonas fluorescens helper bacteria you have listed there are not really known to be helper bacteria. They're known to be plant growth promoting bacteria, but not helper bacteria. So.
Yes, not all of those are expected to be strong beneficiaries in this interaction. They were deliberately selected so you have some neutrals and some negatives. So you're correct, the fluorescent strains were selected to have a range of positive, negative, and different interactions. We do see, let me see if I can get up to this one real quick. For example, just trivial, oh, that's no good. It's your squints in the bottom one. I'm going to ask you to take my word for it because it's completely invisible in this figure. So we're growing, those are little baby aspens, cultured with and without all four of the fluorescent strains on low phosphorus. Yes, low phosphorus. Even some of the ones that are negative, the SW5, which should be a negative one, we actually get better growth on low phosphorus than you get aspen alone. So that's why I'm saying these things are very media dependent. It's the interaction with these things is completely dependent on the media environment. Things that might normally be, if they're not helpful, even if they're not necessarily a microbial interaction in this case, there's a range of possible interaction that these things have that we can identify and use that variability in response to model. As far as using Arabidopsis to model aspen protein-protein interactions, the actual components of the data that we're using is predicted subcellular locations. So you look across all tens of thousands of Arabidopsis predicted interactions, and you say where are they predicted to be localized, and are there pairs of subcellular locations that are particularly enriched for things that are known to interact. So you have things like cytoplasm. Cytoplasm is obvious. Nucleus extracellular is obvious, and that should be disallowed. And then there's, in between those kind of conditions, there's a range where you say that cytoplasm is likely to interact, something localized in cytoplasm is likely to interact with something in the cytoskeleton, but less likely to be interacting with something in the mitochondrial envelope. So using those kind of annotations, I think it's not that these are orthologs to Arabidopsis proteins. These are picking out physical characteristics of plant proteins that make them amenable to some kind of physical interaction.